Resuming on page 47. Enzymes. Proteins perform many roles in the body, including acting as enzymes. An enzyme is a protein catalyst that increases the rate at which a chemical reaction proceeds without the enzyme being permanently changed. The three-dimensional shape of enzymes is critical for their normal function because it determines the structure of the enzyme's active site. According to the lock and key model of enzyme action, a reaction occurs when the reactants, T, bind to the active site, lock, on the enzyme. This view of enzymes and reactants as rigid structures fitting together has been modified by the induced fit model, in which the enzyme is able to slightly change shape and better fit the reactants. The enzyme is like a glove that does not achieve its functional shape until the hand reactants move into place. At the active site, reactants are brought into close proximity. After the reactants combine, they are released from the active site, and the enzyme is capable of catalyzing additional reactions. The activation energy required for a chemical reaction to occur is lowered by enzymes because they orient the reactants toward each other in such a way that it is more likely a chemical reaction will occur. Slight changes in the structure of an enzyme can destroy the ability of the active site to function. Enzymes are very sensitive to change in temperature or pH, which can break the hydrogen bonds within them. As a result, the relationship between amino acids changes, thereby producing a change in the shape that prevents the enzyme from functioning normally. To be functional, some enzymes require additional non-protein substances called cofactors. A cofactor can be an ion, such as magnesium or zinc, or an organic molecule. Cofactors that are organic molecules, such as certain vitamins, may be referred to as coenzymes. Cofactors normally form part of the enzyme's active site and are required to make the enzyme function. Enzymes are highly specific because their active site can bind only to certain reactants. Each enzyme catalyzes a specific chemical reaction and no others. Many different enzymes are therefore needed to catalyze the many chemical reactions of the body. Enzymes often are named by adding the suffix ase, spelled A-S-E, to the name of the molecules on which they act. For example, an enzyme that catalyzes the breakdown of lipids is a lipase, and an enzyme that breaks down proteins is called a protease. Enzymes control the rate at which most chemical reactions proceed in living systems. Consequently, they control essentially all cellular functions. At the same time, the activity of enzymes themselves is regulated by several mechanisms within the cells. Some mechanisms control the enzyme concentration by influencing the rate at which the enzymes are synthesized. Others alter the activity of existing enzymes. Much of what is known about the regulation of cellular activity involves knowledge of how enzyme activity is controlled. Nucleic acids, DNA, and RNA. Deoxyribonucleic acid, DNA, is the genetic material of cells, and copies of DNA are transferred from one generation of cells to the next generation. DNA contains the information that determines the structure of proteins. Ribonucleic acid, RNA, is structurally related to DNA, and three types of RNA also play important roles in protein synthesis. In Chapter 3, the means by which DNA and RNA direct the functions of the cells are described. The nucleic acids are large molecules composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Both DNA and RNA consist of basic building blocks called nucleotides. Each nucleotide is composed of a monosaccharide to which a nitrogenous organic base and a phosphate group, end of page 47, beginning page 48, are attached. The 5-carbon monosaccharide is deoxyribose for DNA. It is ribose for RNA. The nitrogenous organic bases consist of carbon and nitrogen atoms organized into rings. They are bases because the nitrogen atoms tend to take up hydrogen ions from solution. The nitrogenous organic bases are thymine, cytosine, and uracil, which are single-ringed pyrimidines, and adenine, or adenine, and guanine, which are double-ringed purines. DNA has two strands of nucleotides joined together to form a twisted ladder-like structure called a double helix. The uprights of the ladder are formed by covalent bonds between the deoxyribose molecules and phosphate groups of adjacent nucleotides. The rungs of the ladder are formed by the bases of nucleotides of one upright connected to the bases of the other upright by hydrogen bonds. Each nucleotide of DNA contains one of the organic bases: adenine, thymine, cytosine, or guanine. Complementary base pairs are organic bases held together by hydrogen bonds. Adenine and thymine are complementary base pairs because the structure of these organic bases allow two hydrogen bonds to form between them. Cytosine and guanine are complementary base pairs because the structure of these organic bases allow three hydrogen bonds to form between them. The two strands of a DNA molecule are said to be complementary if the sequence of bases in one DNA strand is known. The sequence of bases in the other strand can be predicted because of complementary base pairing. The two nucleotide strands of a DNA molecule are antiparallel, meaning that the two strands lie side by side, but their sugar phosphate, quote, backbones, end quote, extend in opposite directions because of the orientation of their nucleotides. A nucleotide has a 5' end and a 3' end. Now, as a brief note, it's abbreviated by 5' and 3'. back to the reading. The prime sign is used to indicate the carbon atoms of the deoxyribose sugar, which are numbered 1' to 5'. The sequence of organic bases in DNA is a, quote, code, end quote, that stores information used to determine the structures and functions of cells. A sequence of DNA bases that directs the synthesis of proteins or RNA molecules is called a gene. See Chapter 3 for more information on genes. Genes determine the type and sequence of amino acids found in protein molecules. Because enzymes are proteins, DNA structure determines the rate and type of chemical reactions that occur in cells by controlling enzyme structure. The information contained in DNA, therefore, ultimately defines all cellular activities. Other proteins, such as collagen, that are coded by DNA, determine many of the structural features of humans. RNA has a structure similar to a single strand of DNA. Like DNA, 4 different nucleotides make up the RNA molecule, and the organic bases are the same, except that thymine is replaced with uracil. Uracil can bind only to adenine. End of page 48. Page 49 just has different illustrations of 2 nucleotides and a strand of DNA. I will just read the notes, but I will not provide an image. 1. The building blocks of nucleic acids are nucleotides, which consist of a phosphate group, a sugar, and a nitrogen base. 2. The phosphate groups connect the sugars to form 2 strands of nucleotides. 3. Hydrogen bonds between the nucleotides join the 2 nucleotide strands together. Adenine binds to thymine, and cytosine binds to guanine. 4. Deoxyribose carbon atoms are numbered. One end of a DNA strand has a 3' end because of the orientation of its nucleotides. 5. The other end of a DNA strand has a 5' end. 6. The complementary strands are antiparallel in that the 5' to 3' direction of one strand runs counter to the 5' to 3' direction of the other strand. 7. The nucleotide strands coil to form a double-stranded helix. End of page 49, beginning page 50. Top of page 50 has another illustration of adenosine and adenine and ribose and some phosphate groups. There's 3 notes. 1. Adenosine is adenine, which is one of the nitrogenous bases in DNA, combined with the sugar ribose. 2. Adenosine diphosphate, ADP, is adenosine with 2 phosphate groups. 3. Adenosine triphosphate, ATP, is adenosine with 3 phosphates. Back to the main reading. Adenosine triphosphate. Adenosine triphosphate, ATP, is an especially important organic molecule found in all living organisms. It consists of adenosine and 3 phosphate groups. Adenosine is the sugar ribose with the organic base adenine. The potential energy stored in the covalent bond between the second and third phosphate groups is important to living organisms because it provides the energy used in nearly all of the chemical reactions within cells. The catabolism of glucose and other nutrient molecules result in chemical reactions that release energy. Some of that energy is used to synthesize ATP from ATP and an inorganic phosphate group, abbreviated as PI. ATP plus PI plus energy from catabolism equals ATP. The transfer of energy from nutrient molecules to ATP involves a series of oxidation reduction reactions in which a high-energy electron is transferred from one molecule to the next molecule in the series. In Chapter 25, the oxidation reduction reactions of metabolism are considered in greater detail. Once produced, ATP is used to provide energy for other chemical reactions, anabolism, or to drive cell processes such as muscle contraction. In the process, ATP is converted back to ATP and an inorganic phosphate group. ATP is equal to ADP plus inorganic phosphate plus energy for anabolism and other cell processes. ATP is often called the energy currency of cells because it is capable of both storing and providing energy. The concentration of ATP is maintained within a narrow range of values, and essentially all energy requiring chemical reactions stop when there is an inadequate quantity of ATP. Cyanide poisoning. Cyanide compounds can be lethal to humans because they interfere with the production of ATP in mitochondria. See Chapter 25. Without adequate ATP, cells die because there is inadequate energy for anabolic chemical reactions, active transport, and other energy-requiring cell processes. The heart and brain are especially susceptible to cyanide poisoning. Inhalation of smoke released by the burning of rubber and plastic by household fires is the most common cause of cyanide poisoning. Cyanide poisoning by inhalation or absorption through the skin can also occur in certain manufacturing processes, and cyanide gas was used during the Holocaust to kill people. Deliberate suicide by ingesting cyanide is rare, but was made famous by the suicide capsules in spy movies. In 1982, 7 people in the Chicago area died after taking Tylenol that some unknown person had laced with cyanide. Subsequent copycat tamperings occurred and led to the widespread use of tamper-proof capsules and packaging. Summary. Chemistry is the study of the composition, structure, and properties of substances and the reactions they undergo. Much of the structure function of healthy or diseased organisms can be understood at the chemical level. Basic Chemistry, page 24. Matter, mass, and weight. 1. Matter is anything that occupies space. 2. Mass is the amount of matter in an object. 3. Weight results from the force exerted by Earth's gravity on matter. Elements and atoms. 1. An element is the simplest type of matter with unique chemical and physical properties. 2. An atom is the smallest particle of an element that has the chemical characteristics of that element. An element is composed of only one kind of atom. 3. Atoms consist of protons, neutrons, and electrons. Protons are positively charged, electrons are negatively charged, and neutrons have no charge. Protons and neutrons are found in the nucleus. Electrons, which are located around the nucleus, can be represented by an electron cloud. 4. The atomic number is the unique number of protons in an atom. The mass number is the sum of the protons and the neutrons. 5. Isotopes are atoms that have the same atomic number but different mass numbers. 6. The atomic mass of an element is the average mass of its naturally occurring isotopes, weighted according to their abundance. 7. A mole of a substance contains Avogadro's number, 6.22 multiplied by 10 to the 23rd power, of atoms, ions, or molecules. The molar mass of a substance is the mass of 1 mole of the substance, expressed in grams. Electrons and chemical bonding. 1. The chemical behavior of atoms is determined mainly by their outermost electrons. A chemical bond occurs when atoms share or transfer electrons. Brief note before I continue. Page 50 ended at the end of number 3 under the section Elements and Atoms. So right now I'm on page 51. Resuming the reading. 2. Number 2 under section Electrons and Chemical Bonding. Ions are atoms that have gained or lost electrons. An atom that loses 1 or more electrons becomes positively charged and is called a cation. An anion is an atom that becomes negatively charged after accepting 1 or more electrons. Ionic bonding is the attraction of the oppositely charged cation and anion to each other. 3. Covalent bond is the sharing of electron pairs between atoms. A polar covalent bond results when the sharing of electrons is unequal and can produce a polar molecule that is electrically asymmetric. Molecules and compounds. 1. A molecule is 2 or more atoms chemically combined to form a structure that behaves as an independent unit. A compound is 2 or more different types of atoms chemically combined. 2. The kinds and numbers of atoms or ions in a molecule or compound can be represented by a formula consisting of the symbols of the atoms or ions plus subscripts denoting the number of each type of atom or ion. 3. The molecular mass of the molecule or compound can be determined by adding up the atomic mass of its atoms or ions. Intermolecular forces. 1. A hydrogen bond is the weak attraction that occurs between the oppositely charged regions of polar molecules. Hydrogen bonds are important in determining the three-dimensional structure of large molecules. 2. Solubility is the ability of one substance to dissolve in another. Ionic substances that dissolve in water by dissociation are electrolytes. Molecules that do not dissociate are non-electrolytes. Chemical reactions and energy. Page 32. Synthesis reactions. 1. Synthesis reactions are the chemical combination of two or more substances to form a new or larger substance. 2. Dehydration reactions are synthesis reactions in which water is produced. 3. Anabolism is the sum of all the synthesis reactions in the body. Decomposition reactions. 1. Decomposition reactions are the chemical breakdown of a larger substance to two or more different smaller substances. 2. Hydrolysis reactions are decomposition reactions in which water is depleted. 3. All of the decomposition reactions in the body are called catabolism. Reversible reactions. Reversible reactions produce an equilibrium condition in which the amount of reactants relative to the amount of products remain constant. Oxidation reduction reactions. Oxidation reduction reactions involve the complete or partial transfer of electrons between atoms. Energy. 1. Energy is the ability to do work. Potential energy is stored energy, and kinetic energy is energy resulting from movement of an object. 2. Chemical energy. Chemical bonds are a form of potential energy. Chemical reactions in which the products contain more potential energy than the reactants require the input of energy. Chemical reactions in which the products have less potential energy than the reactants release energy. 3. Heat energy. Heat energy is energy that flows between objects that are at different temperatures. Heat energy is released in chemical reactions and is responsible for body temperature. Speed of chemical reactions. 1. Activation energy is the minimum energy that the reactants must have to start a chemical reaction. 2. Enzymes are specialized protein catalysts that lower the activation energy for chemical reactions. Enzymes speed up chemical reactions, but are not consumed or altered in the process. 3. Increased temperature and concentration of reactants can increase the rate of chemical reactions. Inorganic chemistry, page 36. Inorganic chemistry is mostly concerned with non-carbon containing substances, but does include some carbon-containing substances such as carbon dioxide and carbon monoxide. Water. 1. Water is a polar molecule composed of one atom of oxygen and two atoms of hydrogen. 2. Water stabilizes body temperature, protects against friction and trauma, makes chemical reactions possible, directly participates in chemical reactions, example, dehydration and hydrolysis reactions, and is a mixing medium, example, solutions, suspensions, and colloids. 3. A mixture is a combination of two or more substances physically blended together, but not chemically combined. 4. A solution is any liquid, gas, or solid in which the substances are uniformly distributed with no clear boundary between the substances. 5. A solute dissolves in the solvent. 6. A suspension is a mixture containing materials that separate from each other unless they are continually physically blended together. 7. A colloid is a mixture in which a dispersed solute-like substance is distributed throughout a dispersing solvent-like substance. Particles do not settle out of a colloid. End of page 51, beginning page 52. Solution concentrations. 1. One way to describe solution concentration is an osmol, which contains Avogadro's number, 6.022 multiplied by 10 to the 23rd power, of particles. Example, atoms, ions, or molecules in 1 kilogram of water. 2. A milliosmol is 1/1000th of an osmol. Acids and bases. 1. Acids are proton, i.e. hydrogen ion, donors, and bases, e.g. hydroxide ion, are proton acceptors. 2. A strong acid or base almost completely dissociates in water. A weak acid or base partially dissociates. 3. The pH scale is the hydrogen ion concentration in a solution. A neutral solution has an equal number of hydrogen ions and hydroxide ions, and is assigned a pH of 7. Acid solutions, in which the number of hydrogen ions is greater than the number of hydroxide ions, have a pH values less than 7. Basic or alkaline solutions have more hydroxide ions than hydrogen ions, and a pH greater than 7. 4. A salt is a molecule consisting of a cation other than hydrogen ions, and an anion other than hydroxide ions. Salts are formed when acids react with bases. 5. A buffer is a solution of a conjugate acid-base pair that resists changes in pH when acids or bases are added to the solution. Oxygen. Oxygen is necessary in the reactions that extract energy from food molecules in living organisms. Carbon dioxide. During metabolism, when the organic molecules are broken down, carbon dioxide and energy are released. Organic chemistry, page 39. Organic molecules contain carbon atoms bound together by covalent bonds. Carbohydrates. 1. Monosaccharides are the basic building blocks of other carbohydrates. They, especially glucose, are important sources of energy. Examples are ribose, deoxyribose, glucose, fructose, and galactose. 2. Disaccharide molecules are formed by dehydration reactions between two monosaccharides. They are broken apart into monosaccharides by hydrolysis reactions. Examples of disaccharides are sucrose, lactose, and maltose. 3. Polysaccharides are many monosaccharides bound together to form long chains. Examples include cellulose, starch, and glycogen. Lipids. 1. Triglycerides are composed of glycerol and fatty acids. 1, 2, or 3 fatty acids can attach to the glycerol molecule. Fatty acids are straight chains of carbon molecules with a carboxyl group. Fatty acids can be saturated, only single covalent bonds between carbon atoms, or unsaturated, one or more double covalent bonds between carbon atoms. Energy is stored in fats. 2. Phospholipids are lipids in which a fatty acid is replaced by a phosphate-containing molecule. Phospholipids are a major structural component of plasma membranes. 3. Steroids are lipids composed of 4 interconnected ring molecules. Examples include cholesterol, bile salts, and sex hormones. 4. Other lipids include fat-soluble vitamins, prostaglandins, thromboxanes, and leukotrienes. Proteins. 1. The building blocks of protein are amino acids, which are joined by peptide bonds. 2. The number, kind, and arrangement of amino acids determine the primary structure of a protein. Hydrogen bonds between amino acids determine secondary structure, and hydrogen bonds between amino acids and water determine tertiary structure. Interactions between different protein subunits determine quaternary structure. 3. Enzymes are protein catalysts that speed up chemical reactions by lowering their activation energy. 4. The active sites of enzymes bind only to specific reactants. 5. Cofactors are ions or organic molecules such as vitamins that are required for some enzymes to function. Nucleic acids, DNA and RNA. 1. The basic unit of nucleic acids is the nucleotide, which is a monosaccharide with an attached phosphate and organic base. 2. DNA nucleotides contain the monosaccharide deoxyribose and the organic base adenine, thymine, guanine, or cytosine. DNA occurs as a double strand of joined nucleotides. Each strand is complementary and anti-parallel to the other strand. 3. A gene is a sequence of DNA nucleotides that determines the structure of a protein or RNA. 4. RNA nucleotides are composed of the monosaccharide ribose. The organic bases are the same as for DNA, except that thymine is replaced with uracil. Adenosine triphosphate. ATP stores energy derived from catabolism. The energy is released from ATP and is used in anabolism and other cell processes. The rest of page 52 and 53 are review and comprehension multiple choice questions, so I will skip them. Chapter 3, Cell Biology and Genetics. And this is page 55. The cell is the basic structural and functional unit of all living organisms. The characteristic functions of cells include cell metabolism and energy use, synthesis of molecules such as proteins and nucleic acids, communication between cells, reproduction, and inheritance. Cells are like very complex but minute factories that are always active, carrying out the functions of life. These microscopic factories are so small that an average-sized cell is only 1/5 the size of the smallest dot you can make on a sheet of paper with a sharp pencil. Each human body is made up of trillions of cells. If each cell were the size of a standard brick, the colossal human statue made from those bricks would be 6 miles high. All the cells of an individual originate from a single fertilized cell. During development, cell division and specialization give rise to a wide variety of cell types, such as nerve, muscle, bone, fat, and blood cells. Each cell type has important characteristics that are critical to the normal function of the body as a whole. One of the important reasons for maintaining homeostasis is to keep the trillions of cells that form the body functioning normally. This chapter outlines functions of the cell, page 56, how we see cells, page 58, the composition of the plasma membrane, page 58, membrane lipids, page 58, and membrane proteins, page 58. It addresses movement through the plasma membrane, page 64, and endocytosis and exocytosis, page 71. The chapter then addresses the cytoplasm, page 76, and organelles, page 77. It presents genes and gene expression, page 86, and cell life cycle, page 91, and genetics, page 93. End of page 55, beginning page 56. Functions of the cell. Because cells are the basic units of all living things, they are the smallest parts of an organism, such as a human, that have the characteristics of life. Although cells may have quite different structures and functions, they share several characteristics. The plasma, or cell, membrane forms the outer boundary of the cell, through which the cell interacts with its external environment. The nucleus is usually located centrally. It directs cell activities, most of which take place in the cytoplasm, located between the plasma membrane and the nucleus. Within cells, specialized structures called organelles perform specific functions. The characteristic functions of the cell include the following. 1. Cell metabolism and energy use. The chemical reactions that occur within cells are referred to as metabolic reactions and collectively as cell metabolism. The energy released from some metabolic reactions provides the energy necessary for cellular activities, such as the synthesis of molecules and muscle contraction. During some metabolic reactions, energy is released as heat, which helps maintain body temperature. 2. Synthesis of molecules. Cells synthesize various types of molecules, including proteins, nucleic acids, and lipids. The molecules synthesized by the various cells of the body differ. Therefore, the structural and functional characteristics of cells are determined by the types of molecules they produce. 3. Communication. Cells produce and respond to chemical and electrical signals that allow them to communicate with one another. For example, nerve cells produce chemical signals by which they communicate with muscle cells, and muscle cells respond by contracting or relaxing. 4. Reproduction and inheritance. Most cells contain a complete copy of all genetic information of the individual. This genetic information ultimately determines the structural and functional characteristics of the cell. During the growth of an individual, cells divide to produce new cells, each containing the same genetic information. Specialized cells of the body, called gametes or gametes, are responsible for transmitting genetic information to the next generation. And now there's a illustration of a cell and different parts of a cell. A generalized human cell showing the plasma membrane, nucleus, and cytoplasm with its organelles. Although no single cell contains all these organelles, many cells contain a large number of them. End of page 56, beginning page 57, table 3.1, summary of cell parts and functions. Cell part, plasma membrane. Structure, lipid bilayer composed of phospholipids and cholesterol with proteins that extend across or are embedded in either surface of the lipid bilayer. Function, outer boundary of cells that controls entry and exit of substances. Receptor molecules function in intercellular communication. Marker molecules enable cells to recognize one another. Cell part, cytoplasm, cytosol. Fluid part. Structure, water with dissolved ions and molecules. Collide with suspended proteins. Function, contains enzymes that catalyze decomposition and synthesis reactions. ATP is produced in glycolysis reactions. Cell part, cytoskeleton, microtubules. Structure, hollow cylinders composed of the protein tubulin, 25 nm in diameter. Function, support the cytoplasm and form centrioles or centrioles, spindle fibers, cilia, and flagella, responsible for movement of structures in the cell. Cell part, intermediate filaments. Structure, protein fibers, 10 nm in diameter. Function, provide structural support to cells. Cell part, actin filaments. Structure, small fibrils of the protein actin, 8 nm in diameter. Function, provide structural support to cells, support microvilli responsible for cell movements. Cell part, cytoplasmic inclusions. Structure, aggregates of molecules manufactured or ingested by the cell, may be membrane bound. Function, function depends on the molecules, energy storage, lipids, glycogen, oxygen transport, hemoglobin, skin color, melanin, and others. Cell part, organelles, nucleus, nuclear envelope. Structure, double membrane enclosing the nucleus. The outer membrane is continuous with the endoplasmic reticulum. Nuclear pores extend through the nuclear envelope. Function, separates nucleus from cytoplasm and regulates movement of materials into and out of the nucleus. Cell part, chromatin. Structure, dispersed thin strands of DNA, histones, and other proteins. Condenses to form chromosomes during cell division. Function, DNA regulates protein, e.g. enzyme, synthesis, and therefore the chemical reactions of the cell. DNA is genetic or hereditary material. Cell part, nucleolus. Structure, one or more dense bodies consisting of ribosomal RNA and proteins. Function, assembly site of large and small ribosomal subunits. Cell part, cytoplasmic organelles, ribosome. Structure, ribosomal RNA and proteins form large and small subunits attached to endoplasmic reticulum, or free ribosomes are distributed throughout the cytoplasm. Function, site of protein synthesis. Cell part, rough endoplasmic reticulum. Structure, membranous tubules and flattened sacs with attached ribosomes. Function, protein synthesis and transport to Golgi apparatus. Cell part, smooth endoplasmic reticulum. Structure, membranous tubules and flattened sacs with no attached ribosomes. Function, manufactures lipids and carbohydrates, detoxifies harmful chemicals, stores calcium. Cell part, Golgi or Golgi apparatus. Structure, flattened membrane sacs stacked on each other. Function, modifies, packages, and distributes proteins and lipids for secretion or internal use. Cell part, secretory vesicle. Structure, membrane-bound sac pinched off Golgi apparatus. Function, carries proteins and lipids to cell surface for secretion. Cell part, lysosome. Structure, membrane-bound vesicle pinched off Golgi apparatus. Function, contains digestive enzymes. Cell part, peroxisome. Structure, membrane-bound vesicle or vesically. Function, one site of lipid and amino acid degradation. Breaks down hydrogen peroxide. Cell part, proteasomes. Structure, tube-like protein complexes in the cytoplasm. Function, break down proteins in cytoplasm. Cell part, mitochondria. Structure, spherical, rod-shaped, or thread-like structures, enclosed by double membrane. Inner membrane forms projections called cristae. Function, major site of ATP synthesis when oxygen is available. Cell part, centrioles. Structure, pair of cylindrical organelles in the centrosome consisting of triplets of parallel microtubules. Function: centers for microtubule formation. Determine cell polarity during cell division. Form the basal bodies of cilia and flagella. Cell part: spindle fibers. Structure: microtubules extending from the centrosome to chromosomes and other parts of the cell, i.e. aster fibers. Functions: assist in the separation of chromosomes during cell division. Cell part: cilia. Structure: extensions of the plasma membrane containing doublets of parallel microtubules, 10 micrometers in length. Function: move materials over the surface of cells. Cell part: flagellum or flagellum. Structure: extension of the plasma membrane containing doublets of parallel microtubules, 55 micrometers in length. Function: in humans, responsible for movement of spermatoza. Cell parts: microvilli. Structure: extension of the plasma membrane containing microfilaments. Function: increase surface area of the plasma membrane for absorption and secretion, modified to form sensory receptors. End of page 57. Beginning page 58. How we see cells. Most cells are too small to be seen with the unaided eye. As a result, it is necessary to use microscopes to study them. Light microscopes allow us to visualize general features of cells. Electron microscopes, however, must be used to study the fine structure of cells. A scanning electron microscope, SEM, allows us to see the features of the cell surface and the surfaces of internal structures. A transmission electron microscope, TEM, allows us to see, quote, through, end quote, parts of the cell and thus to discover other aspects of cell structure. A more detailed description of microscopes and their use can be found on page 112. Plasma membrane. The plasma membrane is the outermost component of a cell. It functions as a boundary separating the substances inside the cell, which are intracellular, from substances outside the cell, which are extracellular. Sometimes extracellular substances are referred to as intercellular, meaning between cells. The plasma membrane encloses and supports the cell contents. It attaches cells to the extracellular environment or to other cells. The cell's ability to recognize and communicate with each other takes place through the plasma membrane. In addition, the plasma membrane determines what moves into and out of cells. As a result, the intracellular contents of cells is different from the extracellular environment. The regulation of ion movement by cells results in a charge difference across the plasma membrane called the membrane potential. The outside of the plasma membrane is positively charged compared with the inside because there are more positively charged ions immediately on the outside of the plasma membrane and more negatively charged ions and proteins inside. The membrane potential is an important feature of a living cell's normal function. Will be considered in greater detail in chapters 9 and 11. A plasma membrane consists of 45% to 50% lipids, 45% to 50% proteins, and 4 to 8% carbohydrates. The carbohydrates combine with lipids to form glycolipids, and with proteins to form glycoproteins. The glycocalyx is the collection of glycolipids, glycoproteins, and carbohydrates on the outer surface of the plasma membrane. The glycocalyx also contains molecules absorbed from the extracellular environment, so there is often no precise boundary where the plasma membrane ends and the extracellular environment begins. Membrane lipids. The predominant lipids of the plasma membrane are phospholipids and cholesterol. Phospholipids readily assemble to form a lipid bilayer, a double layer of phospholipid molecules, because they have a polar, charged head and a nonpolar, uncharged tail. See Chapter 2. The polar hydrophilic, water-loving heads are exposed to water inside and outside the cell, whereas the nonpolar hydrophobic, water-fearing tails face one another in the interior of the plasma membrane. The modern concept of the plasma membrane, the fluid mosaic model, suggests that the plasma membrane is neither rigid nor static in structure, but is highly flexible and can change its shape and composition through time. The lipid bilayer functions as a liquid in which other molecules, such as proteins, are suspended. The fluid nature of the lipid bilayer has several important consequences. It provides an important means of distributing molecules within the plasma membrane. In addition, slight damage to the membrane can be repaired because the phospholipids tend to reassemble around damaged sites and close them. The fluid nature of the lipid bilayer also enables membranes to fuse with one another. Cholesterol is the other major lipid in the plasma membrane. See Chapter 2. It is interspersed among the phospholipids and accounts for about 1/3 of the total lipids in the plasma membrane. The hydrophilic OH group of cholesterol extends between the phospholipid heads to the hydrophilic surface of the membrane, whereas the hydrophobic part of the cholesterol molecules lies within the hydrophobic region of the phospholipids. The amount of cholesterol in a particular plasma membrane is a major factor in determining the fluid nature of the membrane. Cholesterol limits the movement of phospholipids, providing stability to the membrane. Membrane proteins. The basic structure of the plasma membrane and some of its functions are determined by its lipids, but many functions of the plasma membrane are determined by its proteins. Some protein molecules, called integral or intrinsic proteins, penetrate deeply into the lipid bilayer, in many cases extending from one surface to the other, whereas other proteins, called peripheral or extrinsic proteins, are attached to either the inner or outer surfaces of the lipid bilayer. Integral proteins consist of regions made up of amino acids with hydrophobic R groups and other regions of amino acids with hydrophilic R groups. See Chapter 2. The hydrophobic regions are located within the hydrophobic part of the membrane, and the hydrophilic regions are located at the inner or outer surface of the membrane or line channels through the membrane. Some, end of page 58, beginning page 59. Peripheral proteins may be bound to integral proteins, whereas others are bound to the polar heads of the phospholipid molecules. Membrane proteins can function as marker molecules, attachment proteins, transport proteins, receptor proteins, or enzymes. The ability of membrane proteins to function depends on their three-dimensional shapes and their characteristics. Marker molecules. Marker molecules are cell surface molecules that allow cells to identify one another or other molecules. They are mostly glycoproteins, proteins with attached carbohydrates, or glycolipids, lipids with attached carbohydrates. The protein portions of glycoproteins may be either integral or peripheral proteins. Examples include recognition of an oocyte, spelled oocyte, by a sperm cell, and the ability of the immune system to distinguish between self cells and foreign cells, such as bacteria or donor cells in an organ transplant. Intercellular communication and recognition are important because cells are not isolated entities. They must work together to ensure normal body functions.